non-essential for us. We learned that there are sweet amino acids, also glycine and alanine. And then there are non-protein amino acids like ornithine, citrulline and GABA. Then it is possible for the amino acids to undergo methylation, acetylation, phosphorylation, even to add hydroxyl group, even to develop disulfide bonds. And disulfide bonds are possible only in cysteine. And when two cysteines join together, it is also mentioned as cysteine. Now, which bonds are present? Which bonds are present in the proteins is that amino acids, when joined together, they form amide bond or peptide bond, and this bond causes two amino acids to join to form a dipeptide. If there are equal to or less than 10 amino acids, it is called oligopeptide. If there are more than 20, it is called as polypeptide. Then we learn that proteins are first one because they are the mixed or the heteropolymer, and because they have the greatest diversity around us, that's why they were called as proteins. They can be complete proteins or incomplete proteins. That means complete means all essential, semi-essential amino acids it carries. That means animal proteins. Otherwise, second class protein or incomplete proteins are plant proteins, which may be deficient in one or two essential or semi-essential amino acids. Then proteins can be monomeric or oligomeric. If they carry a single polypeptide chain, monomeric. If they carry two or more chains, oligomeric or multimeric. And examples we learned were insulin, immunoglobulin, hemoglobin, in this case lysozyme and myoglobin. And each individual chain in oligomeric proteins are called as protomeres. Then I gave you the description about insulin, that how many amino acids it carries. Then the heaviest and the lightest amino acid. Then proteins have heaviest and lightest proteins. Then isoelectric point is the pH at which the amino acid will become neutral and it will undergo precipitation. And for example, casein protein undergoes precipitation at 4.7 pH in the curd formation. That's why at the acidic point, the protein present in milk, casein protein, it's a phosphocasein, basically carries phosphate group. And then it is possible that if you change pH or you cause the strong alkali treatment, strong acid treatment or heating, proteins can undergo denaturation. It can be a permanent damage to the bonds, that is called as the permanent denaturation, or it can be a temporary damage which can be restored by proper treatment, and then it will be called as renaturation. We learned the various degrees of the protein structure. Primary structure is the structure made up of chain of amino acid. Number of amino acids are fixed and the sequence is also fixed, and the bond formed is peptide bond. First terminal is called N terminal, last terminal is called the C terminal, and the distance between two peptide bonds will be 0.3 nanometer. Then we learned that secondary structure is also present in proteins. Secondary structure develops because of the intra-polypeptide hydrogen bonds or inter-polypeptide hydrogen bonds. It can be single chain undergoing, so monomeric secondary structure is possible, or it can be oligomeric protein structure also possible. So alpha helical, beta pleated and collagen helix. We learned about the helix breakers are two amino acids, glycines and proline. Then collagen has a different type of structure. We learned about parallel arrangement and anti-parallel arrangement in the case of beta pleated structure. Then tertiary structure develops because of further folding of the secondary structure, and this develops the active sites of the enzymes. And then all other bonds which you learn in chemistry, they all are involved in tertiary structure formation. Finally, this develops the quaternary structure, which is only present in the oligomeric protein. And then we learn the various type of classification possible in proteins. Classification involves, according to shape, they are fibrous, globular and intermediate. According to their functions, they are enzymatic or non-enzymatic, and non-enzymatic has many other types. According to composition, if they carry only amino acids, simple. If they carry a part of amino acid, even they carry some other things like metal or phosphate group or oligosaccharide or nucleic acid or mucopolysaccharide or lipid inside. So accordingly they will be called by different name. Even metals are present there. And derived proteins are the partial breakdown of the proteins will cause formation of proteins, and those proteins are called as coagulated proteins, proteose, peptones and peptides, and this we have already done. Please remember one thing, fibrous protein have only primary structure and secondary structure, no tertiary structure or very little tertiary structure. Then monomeric globular proteins will carry primary, secondary and tertiary structure, and only the oligomeric proteins will have primary, secondary, tertiary as well as quaternary structure. So we will start with the new topic of nucleic acid and its components in the next class.